Hey, everybody, and thank you for joining us. Welcome to Post-Hoc Live. I'm Drew Armstrong, the executive editor of Endpoints News and your host today. We've got an interview that I'm really excited about doing, a conversation that I have wanted to have for a long time coming up today. I will tell you who that is going to be in a moment, but just a little housekeeping to get us started. If you are joining us for the first time, welcome and hello. We bring you the reporting, the analysis, and the debate that often happens behind the scenes in our newsroom live to you. If you're with us on YouTube, you're not already following us, please hit that little subscribe button on the bottom of our feed to get alerts about future live streams. You can also sign up for the Post-Hoc email on your Endpoints News subscription page. And if you missed the show, this is going to live on Spotify and Apple Podcasts, as well as on our YouTube channel. So today's guest is a chemist, columnist, and blogger, if I dare say so, an advisor to biotechs, works in the industry, and he writes the In the Pipeline blog on science. His columns I think of as a must read for anybody who cares about biopharma. I assign them to young reporters who join us, as well as experienced reporters and myself. I read most of what he writes. He's going to join our biopharma correspondent, Andy Dunn, and I to talk about his career and his perspectives. He otherwise needs no introduction. So Derek Lowe, welcome to Post-Hoc Live. Thank you so much for being with us. I just wanted to ask you to talk a little bit about your career as a start, because you have this kind of like multi-hyphenate career path where, you know, you're, I believe, a chemist by training, and you worked in the industry, and then you decided that you were going to start writing.
Yeah.
And could you just talk about like what made you decide you wanted to start writing about this industry, like how that happened? Because I understand how it happened for me, but I don't tell us how that began.
Yeah, no, it's a fair question because it's kind of an odd thing to do. I always, even going back and being in school and the like, I was always very quick to put together presentations or papers, things like that. But that's not really a rate limiting step in industrial drug discovery. I mean, it doesn't hurt, but it doesn't make you discover drugs any faster or really that much better. So back in about 2001, I was reading some of the early blogs and I thought, I'd like to do this. And I realized there was no barrier to entry, really. And I thought, what should I write about? And I realized that whenever people asked me what I did for a living and I told them about drug discovery, they're like, that's interesting. I didn't know anyone actually did that. So I thought, there's my subject. People seem interested in it. No one seems to know jack about it. So I should write about that. But when I go back and look, I mean, even back in college, graduate school, my postdoc, I can see these things. I did my postdoc in Germany, and I would write up these long letters about what things were like over in Germany and what, all the sorts of things that were going on. And I would send them, I would copy them out on a copier and send them out to a mailing list of, a dozen or 2 dozen people. And looking back, I was blogging then. So it's something I've always enjoyed doing. I like the writing. I like the speaking about it. So I'm just glad I got the opportunity to sort of merge these things.
How do you decide what you want to write about? Because I think one of the things that I enjoy reading you for is that, as someone who does not have a a science education. I mean, I'm fortunate enough to work here and talk to smart people all day
Yeah,
and read and write about this stuff, but I don't come from a technical background. One day you'll be writing about something going on with Sarepta or with
yeah,
RFK, and the next day you're writing about something in like the material science or, the molecular
yeah,
filters or things like, I mean, how do you pick out what you get interested in and think is worth writing about? Because it's been
yeah.
reasonably varied kind of all along, which I think is particularly fascinating.
It's by choice. I tried deliberately not to write about similar topics on consecutive days because I figure that way, if people get used to that, then they come in and they start reading and say, you know what, I don't care about this stuff. They at least know that the next day is going to be different. They can come back and check it out then. What I have is to write this, I have a pretty big RSS feed of journals, and I scroll through those constantly. And many of them are related to the work I do in the day job of drug discovery. And some of them are a little bit more general scientific spread, science, nature, PNS, and stuff like that. And I just see what catches my eye. And I drop it I have a literature manager program. I use Zotero, and I drop it into a folder called blog fodder, and it grabs the PDF
Yeah.
and drops it in there. And then when it comes time to write a blog post, I'll have several tabs open of things that, I really should write about this. And I'll just start in and see if it takes off. Sometimes it turns out I've taken on a topic that needs more work than I can give it at the time, and I have to circle back around. But most of the time, it's just like, okay, this is something different. Let's talk about this today.
Who do you like when you think about who you're writing
Oh,
for?
yeah.
I mean, because you managed to bridge this gap between, I think, quite sophisticated and often technical, but also educational. I mean, I think, who do you think about as your audience? I mean,
Yeah.
who are you trying to reach? And I'm curious how that conversation with audience has gone over the years, kind of as you've been
Yeah.
doing this for a while.
I mean, at first, I didn't know who my audience was at all. I was just writing into the void and hoping people would show up. I can actually remember the first time that my little traffic counter thing on the old Blogspot site actually showed someone that I didn't know who the hell it was. I was like, whoa, look at that. And over the years, though, I always think of people who, of course, are in the industry, and want to hear about this stuff. But I keep in mind that there are people who aren't, some of them who are not even in the sciences. And that's why on the post, you'll see me break into little italicized paragraphs of background. And I warn people, so, okay, next couple of paragraphs of background. If you know this stuff, skip on. If you don't, read the next two, see if you can. And
Yeah.
it seems to work. Someone stopped me the other day on the commuter rail up here in Massachusetts and said, hey, are you Derek Lowe? I said, yeah. I am. And it turns out this guy reads the blog. He is not a chemist at all. He's over in tech. He does like programming type stuff, but he has no chemistry background. But he says, I enjoy reading your stuff because I can follow a fair amount of it. I said, well, this makes my day because this is what I'm aiming for.
Does that happen to you more now where people recognize you? I mean, is that weird for you?
Ha. It happens in Cambridge and Boston, that's for sure. And I can remember a few times. I can remember once it happening at a burrito place here in the town where I live, and my teenage daughter was with me, and I thought she was going to fall through the floor toward the center of the earth in waves of embarrassment. But yeah, it does happen. And if I go to a big conference or something, if I go to like an American Chemical Society meeting, um, I'm not going to. It was weird the first time it happened. I'm not going to be able to make it across the exhibition floor without people stopping me about every 10 or 12 feet. So, yeah, that's a little strange, but fortunately, that does not carry over to most aspects of life. I can go to the grocery store in safety.
Has that, I'm wondering the flip side of that quickly. Has the writing sort of, particularly for a general audience, has
Yeah,
that made you a better chemist in any way? As far as so much of it is so
yeah.
specialized if you're in the industry.
It has forced me to keep up with the literature to a much more to a wider extent than I ever would have on my own. And I know that for sure. I'm honest with myself about that. You know what the literature is like. It's just this constant fire hose blasting in every direction. And you're like, do I really want to stick my head into that and take a drink? But it has forced me and it's gotten me to read much more widely than I'm pretty sure I would have otherwise. So yeah, it really has helped.
And you do advisory work now. I mean, you're on the board of a AI company, and Andy has written about this.
Yeah,
How did that come about?
yeah. I'm on a couple of SABs, and they approached me. And sometimes it's because of things I'd written on the blog or just the blog in general. They say, "Hey, you seem like an interesting generalist kind of person. We'd like to get your opinions on stuff." So yeah, I'm glad to take those sorts of things on, and it's something I'll probably do more of after I retire from the day job in a few years.
Mhm. Um, can I ask you, kind of going back to these choices about what you write about? I think
Yeah,
one of the things that I and a lot of other people have followed on quite closely is you have, I think, a nose for scientific and regulatory controversy. You've had a lot to say about
yeah,
Sarepta. And can you walk me through kind of when you think about weighing in something like that,
yeah.
how you choose it? And then I think also I would love to get your thought process on the Sarepta
Sure.
writing that you've done, because I think you've probably been one of the kind of the clearest and most forceful voices on like what is a really
Hmm.
complex story for a lot of people.
Yeah, yeah,
And so how do you decide when to wade in and things like that? And then this one in particular.
yeah. With Sarepta, my blog posts have been pretty much timed to FDA decisions and to big clinical news. And that's when I jump in on a lot of topics because there are a lot of things you could write about all the time, but I don't want to have too many posts where it's, okay, this is going on and we don't know what's going to happen, but here's some of the stuff. I want to have a bit more of a hook for, okay, the FDA just said this. What does this mean? Or here are these clinical trial results that just came out. So that's the immediate tie for most of the blog posts. As far as Sarepta and some of the other regulatory decisions that I've written about, I don't know. I feel strange about this because it doesn't seem like to me like I'm saying anything controversial. Apparently I
Mhm.
am. But I think, and I've always thought that the FDA's job is to balance efficacy and safety. That's it. They don't think about prices, of course. That's not their mandate. Efficacy and safety. Is this thing efficacious enough for its safety record? Is its safety record good enough for its efficacy? And I've seen some decisions that really seem to have skewed that, where you have things like some of the Sarepta drugs, and I would have to say some of the anti-amyloid antibodies that have been approved, where the efficacy seems questionable or at the very best not proven or not yet proven by a long shot. And then you wait and wait and wait for long post-marketing studies to be done. And I do not want to go around accusing people in bad faith, but these post-marketing studies do not ever seem to be done very expeditiously
Yeah.
because after all, you're still selling the drug. And then it comes out that in Sarepta's case, maybe it doesn't work. Hmm, interesting. Are they going to withdraw the drug? Well, the FDA has slapped a box on one of theirs, and they're still, as far as I know, selling the products. I can't make it out. Now, in this current environment under the Trump administration, I don't expect to be able to understand anything the FDA is doing because they don't understand what they're doing anymore.
Where do you feel like, can you talk about like, I think in particular with Sarepta or the FDA right now, kind of where you feel like the need to weigh in on something like this? Did you feel like there was like a lack of voice around this in particular, where
Yeah,
you felt like there was really something to say that nobody was saying? And I think I asked that in the context because
yeah,
I think people who sit in my job, I think we're covering the news and we're writing about the news. And I think obviously sometimes that does come with a certain perspective, and you can
yeah,
see that in how we cover things. But I think also, a lot of times we try to restrain some of that perspective in
yeah.
order to try and do the news, and you're not
No,
constrained by that. Did you feel like
uh-huh.
there was a hole there that needed to be filled?
I suppose so, because yeah, I am, when you get down to it, an opinion writer. Now, I write opinions about a lot of pretty technical subjects, but what you're getting on the blog are my own opinions on that. And I've never, represented them as anything more, anything less. I am not under an obligation to cover the constant stream of news, which is good because by golly, it's pretty constant, isn't it? I pick the things that I think are interesting to me, that I think might be interesting to the people that I imagine reading the website, or that illustrate, I guess, particular scientific or regulatory points that I think should be emphasized. So that's what leads me to
Yeah.
weigh in on this. And something like Sarepta, I'd written about it several times over the years, so I really had to cover the latest stuff and say, look, here's the situation we find ourselves in. How did we get here?
I want to ask you a bit more about your career in chemistry. I mean, you've been doing this for a long time.
Yeah,
What are the things that are most different now? And I ask this in the context of someone who sits on some of these SABs, and you're kind of at
yeah.
the forefront of some of this stuff. What's changed the most, since you
Mm.
started doing this?
Yeah, I think about that some because my first industrial job with Shearing Plow, a company that doesn't exist anymore, was oof, coming up, what are we, 20, well, 36 years ago now. It's in late 89. So a lot of stuff has changed. One thing that really hasn't is a lot of the ways that we do the actual chemistry in the lab. Some things have. We used to run thin layer chromatography plates all the time, and now we use LC mass spec. There are things like that, but we still are weighing out compounds, putting them into glass vessels, pouring in solvent, heating them up, checking the progress, cleaning them up. That sort of thing is still there. There have not been any magic revolutionary advances in how chemistry itself gets done. We're we have a lot more reactions. We have a lot more ways to monitor what's going on, but it's still mix it together and heat it up and see if you get the right product. Now, that said, there are a lot of other things that have changed. Biology, of course, has changed just beyond recognition over that time because of the advent of all the molecular biology techniques. Another thing that has changed, though, I think, is chemical biology, the idea of applying chemical techniques and chemical thinking, a chemical Weltanschauung, if you want to get fancy, to biological systems. That was kind of an exotic thought for a long time, but now I think that a lot of medicinal chemists really have gradually morphed into more chemical biology types because we're able to do all these experiments on
Mhm.
proteins and on cells that were previously just not even thinkable. And we can do all these strange hybrids of small molecules and biological systems that really can tell you a lot and are things that we've never had the opportunity to do. So that has changed. Chemical biology has sort of taken over.
Yeah, and I think we'll definitely get into that as well. I think in the context of where small molecules are headed broadly as a therapeutic modality. But you mentioned, and I think I've talked to many people that would characterize what they're working on as these magical revolutions in drug discovery under
Oh,
the
yeah.
umbrella term of AI. We've talked about this before, Derek, many times. But I want to start maybe to ground the audience in your perspective overall, which I think you've summed up neatly with short-term skeptic, long-term optimist. If
Yeah,
well, one, if that's still true today, and if
it
unpack that
is.
for us as far as how you view AI and biotech
It
broadly.
is. I have this reputation as an AI skeptic, but I would think I'm more of an AI realist based on the sorts of hype that get thrown around. I think that right now, AI, and let me say right off that most of the time when people say AI, they actually mean machine learning or ML. I think that its ability to help us out in the whole drug discovery process is almost inversely proportional to the cost and importance of the questions it's helping us out with. By that, I mean it can help us out quite a bit in the very earliest stages. AlphaFold, for example, is a great advance. That Rosetta Fold type stuff, it's a great advance. But I have never worked on a project whose success or failure was predicated on knowing a protein structure. I mean, it's nice and it definitely can help, but it's not the deal breaker. So you can do a lot of things with that. and you can do a lot of stuff on searching the chemical and biological literature and maybe even getting some ideas on reaction design and design of experiments. But the real problems in the drug industry all go back to our 85% failure rate in the clinic. That's the central fact of biopharma, as far as I'm concerned. Everything that seems weird or inexplicable or impossibly annoying about biopharma comes back to that 85% failure rate. And it's because we pick the wrong targets and we fail in phase 2, or we get some unexpected tox and we fail in phase 3. And AI is not yet able to help us out with either one of those.
And this whole movement of kind of thinking about, at least maybe to focus on the preclinical aspects,
True.
and in the chemistry setting in particular, I hear a lot about this desire to move from artisan kind of bespoke processes that sort of rely on the latent knowledge of human chemists
Yeah.
into something that engineering is often a word that comes up time and again. What do you make of that overall? Is that an accomplishable goal? Is that even a desirable goal?
It's accomplishable, I suppose, but the problem is that organic synthesis has got a huge number of variables in it. So many starting materials, so many reagents, solvents, reaction conditions, that it's been very, very hard to reduce to an engineering problem. Now, in process chemistry and manufacturing, you can reduce specific organic synthetic pathways to that, to the point of sometimes in industry, people will actually build a particular machine, like some sort of flow chemistry apparatus, to do this one reaction that makes this valuable thing happen for this valuable product. And that is engineering all the way. But that's when you've narrowed down. Back in the exploratory part and preclinical, you're doing so many different things, so many different ways, that's been very hard. Now, because of the range of metal catalyzed reactions and other things, we're able to go through a lot more reactions more quickly than we used to if we get into a chemical manifold like that. But that all comes to a screeching halt if you come to a structure that has unusual features in it. And then it's right back to the stare out the window or stare at the whiteboard. And how are we going to do this?
What's your reaction when I'm sure you see some of this stuff when like the head of Microsoft says like we're gonna go from 12 years to 1 year, and in bringing drugs to market. These kind of this very, I would say, tech attitude around
Yes.
solving problems. I mean, does that drive you nuts when you hear
Yes.
that stuff?
In short, it does.
Yeah.
Because these statements land very poorly with people who have actually done drug discovery. And it's not because we're a bunch of old stick in the muds who need to be disrupted either. There are irreducibly complex things in drug discovery, and the first one of those is human biology. Tech people tend to get this attitude, and I see where it comes from. I mean, many of these folks have actually written lines of code or produced pieces of hardware that really have changed the world and the human environment and human thinking. That is going to give you an exaggerated idea of your ability to change the world.
Yeah.
And this is, I think, the root of the problem, because if you're blinded by this, then you look at human biology, you say, oh, I know this. DNA is like machine code. And transcription, that's kind of like a compiler. Yeah, I know how this works. No, you don't. All of the things that Silicon Valley has done have been done with human designed systems, human designed chips, human designed software. Biology is the opposite. It's this absolutely alien tangle of evolutionary legacy, 4 billion years of, hey man, whatever works. And it's just not comprehensible from an engineering standpoint. It'll drive you nuts. You look at it and say, why does the optic nerve make a blind spot in the center of the vision sensing retina? Jesus, that makes no sense. All kinds of stuff like that.
Is
Yeah,
there on the long-term optimism side
Yeah.
of how much do you subscribe to kind of to sort of crib off Demis Hassabis's view of the world, which is that sort of AI, machine learning, these pattern recognition models, that is the right language for biology in the way that math is for some physical problems. I
Hmm.
think that's basically what he was saying at some
Yeah.
point. But that idea of, instead of reductionist thinking to wrap our human brains around of sort of here's a pathway and here's a cartoon diagram of it, it's more embracing exactly that complexity in a way where
Yeah.
maybe we sacrifice interpretability and we don't necessarily understand in the way future exactly
Yeah.
how it's unraveling human biology, but how do
I
you think
think
about that
that
overall?
in one sense, I think he's on to something because I have a talk I give where I show people a blown up bunch of little blobs of color and ask them to analyze it. And we start thinking, okay, X, Y axis, are there patterns in the hues or the size or the shape of these bits of color on the various, you can do all these analyses. The next slide shows what it actually is. It's a very zoomed in portion of Georges Seurat's La Grande Jatte pointillist painting. And I said, no matter how much work you do on that little section of it, you cannot figure out that it's part of this. And the next part of the slide shows a huge tangle of biochemical pathways going to a fluorescently labeled living cell. And you can't go from that to that either. These things are parts of much larger systems with emergent properties. So, in that sense, I think he's right. Where he and I part company, most likely, I have not had the chance to engage him on this subject, but where he and I part company is the idea that we have enough data to do pattern recognition in biology. We don't.
Yeah, I think that's the kind of the attitude we see is, and this is kind of happening all throughout AI and tech right now. Just well, if we have limitless resources, we'll solve this problem, and that's where we see all the capital going
Yep.
into it now.
Yep.
I mean, can I, in the context of this though, how would you imagine? I mean, how do you, when you think about what the future is, you mentioned,
Yeah.
when we were talking that like you're, kind of more towards the end of your career than your beginning.
Yeah.
If we had put you 25-year-old you in a time machine, you were starting
Yeah.
now
Hmm.
with all the knowledge you have, how do you think the job of the medicinal chemist is going to be different in 10 years or
Yeah.
20 years? And what's that workforce going to look like? When you think about kind of the future of what you do, what is that? How do you
Yeah.
consider that?
I had thought of that. It's a really good question. I think that there's still going to be a place for small molecules and small molecule synthesis. We have been trying to get rid of that for decades. And sometimes literally. I mean, a lot of the advances that came along during my career were looked at from one perspective, an attempt to get rid of the medicinal chemists because we are expensive. We are a regulatory burden. We create horrendous waste streams. We're a safety hazard too. Let's be honest. A lot of flammable stuff in there. And the feeling was when the computational boom hit in the late 80s, early 90s, oh, thank God, we're just going to be able to compute our way through. Ooh, okay, maybe not. Then combinatorial chemistry. Oh, okay. We're just going to use these tiny little, robot arms, and we're going to make zillions of compounds. We'll just screen all those, and we'll find our answers there. Okay, maybe not. And there have been all these attempts to sort of get away from this. I don't think it's going to be easy to get away from it. At the same time, I would want my newly time transported 25-year-old me to really embrace the chem bio end of it and embrace the biology. Because a direct example would be things like antibody drug conjugates. Here you have a small molecule made by chemists, which is stuck onto an antibody made by biology. Now it's stuck on there by means that the chemists have had to dream up for the most part, all sorts of linking chains and stuff, which are quite important. I think that's where the future is sort of a marriage of molecular biology and chemistry. In the same way, I think a lot of medicinal chemists have become chemical biologists to some extent. I think a lot of molecular biologists have slowly become more like chemists because they think less in terms of abstracts, like this blob, curvy arrow goes to this triangle, which is influenced by this purple square. Now they're thinking of their species as real molecules with real sizes and shapes and charge distributions and conformations and properties. Well, guess what? You start thinking like that, I'm going to start calling you a chemist.
And
So I
is
would
there
tell,
something,
was
yeah,
that?
is there something special going on right now in the small molecule field? I think we're just putting together, seeing something like Merck's result with their PCSK9, which is a
Yeah,
macrocyclic peptide. From my understanding, just kind of an extraordinary feat in chemistry to
it
make that
is.
reality.
It is.
But what's you have a much broader aperture here. What's going on? Does this amount to something like a renaissance for small molecules right now?
Yeah, it might. And it means that the small molecules are not so small
Yeah.
because you have these large macrocycles and you have these things like the bifunctionals, like the protein degraders, the bifunctional degraders. If you had proposed something like targeted protein degradation and bifunctionals back when I started in the industry, you'd have been fired. You'd have been fired. People would have laughed at you. They said, good God, you think you can just take two proteins and somehow just kind of simple-mindedly bridge them together with like a polyethylene glycol, and this protein A is going to act on protein B even though it's never seen it before? You really think that? Yeah, sure. What's the protein you're going to bring in? Oh, it's going to be an E3 ligase. Oh, that goody. Well, what attracts E3 ligases? Oh, you'll never believe it. Thalidomide. Oh, they'd have fired you. Oh, and this thing you're
Yeah,
making, how
you can't
big?
work in the industry off that idea. Yeah, yeah.
Yeah, how big is this thing going to be? It's going to weigh like 1,200. Yeah, it's great, man. No, they would have fired you. So yeah, I think that what's happened is we finally got out of the old rule of five mindset that ran things back in the early 2000s, where for those who don't know that one, that was a paper done by Chris Lipinski at Pfizer, who analyzed the existing approved drugs and found that they mostly fell into a manifold with a certain number of hydrogen bonds and molecular weight and hydrogen bond acceptors and donors and et cetera, et cetera. And he reduced it to a little mnemonic of no more than five this, no more than five that. That was a disaster in a way because it was so simple the upper management could understand it. And they immediately said, oh my God, finally, we have the key. None of you people make anything outside those rules. You hear me? You're going to get an electric shock through your keyboard if you try to register a compound that's outside the rule of five. And that's just not the way to do it. I mean, things like Merck's molecule for PCSK9, a lot of the protein inhibitors, the bifunctionals, they violate the rule of five backwards, forwards, and sideways.
Do you think that's starting to open people's minds to other possibilities when you kind of think about where that future heads?
It
Are
should.
we breaking away from some of those dogmas?
Absolutely. Absolutely. And it doesn't mean that developing these molecules is easy. They do have a higher failure rate than things that weigh 325 and have a nice log D. But their failure rate is nowhere near zero, and the targets you can approach with these things are worth so much to unmet medical need that you have to do it. So yeah, I think it really has been liberating to see these sorts of things.
I
And
want to,
Art, just
yeah.
because you have again such a wide view of what's going on in the space, what are you obsessed with right now in terms of that?
Hmm.
I think that's an example of what we have seen work. The PCSK9 results, a lot of the molecular glue data, you can
Yeah,
see it playing out in the clinic, I think, as hoped.
yeah.
What's on the edge right now for you of
Hmm.
ideas that are
Oh
kind of
boy.
out there that have your interest that we should be watching, over the next few years? Maybe they work, maybe they don't, but they at
Yeah.
least will
Well, I
challenge
mean,
some
some of
dogmas.
that stuff is stuff I'm doing in the lab, and I can't talk about it. But because I really do like working on exactly the sorts of things you're talking about, the really out there stuff that might actually work. Who knows? I'm going to veer off for a second because I have a theory that I've told many people about that why I like projects like that. I think that management looks down on the portfolio of projects and they say, okay, let's rank these things. That one over there, that project is gold. That thing's just about to go to the clinic. That's going to work. That's going to be a big product. That's going to make all of our bonus targets. I love that project. Then it goes all the way down to this one over here. Oh, geez. Oh, no, man. That's I don't even think that can possibly work. No one's ever gotten stuff like that to work. Yeah, good luck, guys. So you'd think that the risk reward would put you toward the golden big project that everyone's paying attention to, but no, those projects fail pretty often. And they fail less often than the crazy wild-eyed ideas, but they fail a lot more often than you'd like. And when they fail, someone is to blame. Someone's,
Avoid that at all. That's good career advice.
oh, buddy, whatever hits the fan does not get evenly distributed. Believe me, big chunks go flying off. So, meanwhile, over on the crazy squirrely weirdo project, if it fails, nobody cares. Everyone knew it was going to fail. If the big wonder project works, well, everyone knew that was going to work. We already had that penciled in. Meanwhile, if the crazy project works, everyone involved is a hero. So I think the risk reward is actually better over there. Now, this might not be good advice for my 25-year-old self, but as you get a little bit more secure in your career, I think it's definitely worth considering.
Yeah. Work on the weird stuff. Got it. Yeah.
Yeah. I want to ask you one more 25-year-old self question kind of to wrap us up. I mean, would you be telling 25-year-old you to move to China and work in the industry there? Because that's been such a fascinating aspect of this industry right now.
Oh, God. I mean, the history of China has been pretty fraught because we went from having no contact at all to then there's this whole craze of opening up research institutes over there. And partly that was a political and business move. Then there's the whole wave of closing all those research institutes over there.
Yeah.
And now the Chinese industry itself is kind of getting up speed. I don't know what to make of that. I don't know if I would tell myself to move to China, but then I would not. The problem I would have with bringing my 25-year-old self into the year 2025 is I would first have to catch him up on the politics. And I'd say, sit down. You're not going to believe this shit.
Yeah, you've written a lot about that. I mean, you're not shy about waiting in there.
I'm
And
not.
can I ask you, I mean, I talked to a lot of people about this, just to wrap us up here. Everybody I think has got their personal feelings about, I mean, if we can put away the obvious, very strong feelings people have about, democracy and all of these like absolutely massive,
Right, other such
just,
trivial
just,
topic.
yeah, just narrowing in directly about like what is happening to the medical research and funding apparatus
Sure.
in this country and kind of so I used to work in not a lot of people know this about me, but like I used to work in classical music. I worked in a
Oh,
classical music festival
oh,
in
wow.
southern Vermont whose founders were all German and European musicians who had fled World War II and
Mm,
the Nazi regime there
yeah.
and had created this unbelievable nexus of classical music talent in New England.
Hmm.
And their presence in a lot of ways is reflective of the scientific talent that we saw
Yes.
built in this country and that we have invested in for decades and decades and decades in many ways because of that. Are you worried that we are at risk of losing and those type of empires crumble slowly, but like how much does that worry you when you look back at the kind of legacy
Yeah.
you have in this people you've worked with, I presume.
Yeah.
How much of a concern is that for you?
A huge, terrible concern. You ask what's happening to the scientific funding and research system in the US, which I think was honestly the envy of the world. It is being destroyed deliberately by literal barbarians. That's what I think. We are doing terrible things to the economic engine of this country, which has been science and technology innovation. And we're doing it deliberately with malice aforethought in the service of some extremely ignoble goals. It is a horrible thing to witness. And the best I can hope for is that the rest of my life will be spent helping to undo some of this damage. I'm extraordinarily angry that this came along and that it came along when it did. I was going to have a quiet retirement enjoying myself.
Well, I do think your writing is important in this, and I think you've, provided a real voice
Well, thanks.
for your perspective on that. And I think for also just educating people, even if you work in this industry, but maybe don't come from a technical background like me. I
Yeah.
want to, thank you for, I am a much smarter asker of questions and journalist because you're writing. And
Oh,
I
thank
know
you.
there are a lot of people out there who can say the same. And I'm glad we could have you as a part of this conversation today. And I hope we have you back sometime.
Please do. Please
All
do.
right.
I've got a lot more opinions where those came from.
I look forward to hearing them. So, well, that is all the time for today. Derek, Andy, thank you so much for joining us. We are going to be following along with all of this closely as always, and you can keep up with our coverage on endpoints.news or by subscribing to our newsletters. The recording of this conversation is on YouTube, and audio will also be up on Spotify and Apple Podcasts later. A huge thank you to our producer and to our engineer, Julia Harris. We will see you the next time news breaks. For Endpoints News Post-Hoc Live, I am Drew Armstrong.